Hello everyone, this is my second real attempt at getting my hands dirty with gene editing and benchling. And so for this experiment, what I wanted to do was create a single guide RNA that can be used in CRISPR. Before I go deeper into how I was able to complete this experiment, I'd like to fully disclose that I didn't create this experiment or the procedure of it. This was again just off a basis of YouTube, and I'll link it in the description below. And so the first step that I did was I found a human gene, and this human gene is SCO1. And after finding a human gene, what I did is I found the exons.
Just looking over the video right now, and I realized I forgot to explain what an exon really is. An exon is a part of the DNA sequence that has coding information that will allow the DNA to be transcribed into proteins. Anyways, along with the video.
That I would like to take the single guide RNA from. And so when looking at the exons, we can see that we have four right here earlier on in the gene. But what I chose to do was I chose to actually focus on this exon. And this is because it comes after these four exons, meaning that a change in this exon would result in changes in these exons, which is a lot more meaningful of an experiment, and meaning it's a lot more meaningful of a single guide RNA that can be used in CRISPR. It's also important to note, the YouTube video talked about that it has to be away from the 5-UTR region. So we couldn't really choose this exon as well. The next thing that I did was I asked benchling really to that I'm going to create a single guide RNA for CRISPR. And so I asked them to create some potential guide RNAs that I can use. And so when looking at these guide RNAs that could potentially be used by myself, the things that are most important to look at first are the on-target scores and the off-target scores. And here it even tells you it's the optimized score from an article, and it just tells you how it's optimized with PAMs. And so basically what you want from the on-target and off-target scores is you want them to be higher. It goes from a range from 0 to 100 for each respectively. And so the closer it is to 100, the better and more easier it's going to be used as a single guide RNA. And so what I did is I ordered it in a descending order of the on-target score. But when we look at things like this sequence, if you click on it, it's going to take you to where exactly it is in the gene. And the problem with this is it's nowhere near the transcripts, and the transcripts can be found right here. And so if we were to actually take a single guide RNA from this area, it's not going to affect how this DNA can actually produce proteins, which is called the process of transcription. And so it was important that we choose something that actually touches on this area where the transcripts are located. And so the best sequence that I was able to find is in the position 2583. And so here you can see that it touched on the transcripts. And so if we were to use this for a single guide RNA, and then later on to be used in CRISPR, it would actually affect the transcripts, meaning that it's going to affect how this appears as protein. And so I was able to use this sequence to develop the CRISPR single guide RNA. So if we come here, if I were to actually go deeper into this experiment, I was actually to order this, I have the bases here, and so I can send that to a company and then get a real life version of this sequence and use it as a single guide RNA for CRISPR. Overall, this was a great project that I was able to complete. It was very interesting to be able to use benchling once again. And it was a great opportunity to continue to develop my skills with CRISPR. In the past experiment, I used restriction enzymes, but I feel the future of gene editing is really focused around CRISPR. And so it was nice to be able to develop a single guide RNA for that. Once again, if you have any questions or feedback for me, please don't be afraid to put them in the comments section below. And hopefully I'll see you again with another experiment that I can create. Thank you.